Against all these threats and challenges to life, constant progress of research in life science and tight collaboration in relevant industry chains are essential for achieving better vital quality. Originated in 2002, RWD has been constantly furnishing customers with reliable solutions and services in life science, animal health and clinical medicine, and dedicated for better vital quality within our wits and powers. Our products have been sold to over 100 countries and regions around the globe and won worldwide customer confidence and support. At RWD, we launched 6 solutions in the fields of life sciences, animal health, and clinical medicine at the forefront of the world, including animal surgery and modeling solutions, which ensure experiment efficiency through diverse animal disease modeling and model evaluation techniques. In vivo imaging solutions, which provide tangible tech support in fields such as biology, pharmacology, materiology, and translational medicine, among others, by accurately showing blood flow and changes in other indicators of live beings in visualized imaging. Neurosignal research solutions, which are valuable tools for neural circuit analysis, as well as research into mechanisms for neural system diseases, development of brain-machine intelligence technology, and more. Histopathology solutions, which improve the quality and efficiency of pathological diagnosis while sectioning with high accuracy and efficiency. Cellular and molecular biology research solutions, which advance efficient scientific research through covering all conventional experiments, including cell separation and purification, self-culturing, self-analysis, and molecular biology. Animal diagnosis and treatment solutions, which evaluate diagnostic efficiency and treatment efficacy of surgery with anesthesia, dental services, rehabilitation, and physiotherapy, urinary system diseases, etc. And our assistants, science researchers, have published thousands of papers in a number of world-leading academic journals, including Nature and Science. On top of that, many scientific research institutions, medical institutions, and pet diagnosis and treatment centers have attained their success by leveraging our 6 solutions up to now. We will maintain cooperation and innovation with an open mind, boosting growth within many industries through joint efforts with our partners.
So we've been collaborating and cooperating with RWD for quite some time now, and their self-developed products have helped us in the laboratory maintain accuracy and improve accuracy and help our efficiency. We've used everything from cryostats for sectioning various organs of the body, all the way through to stereotactic surgery solutions to deliver small amounts of virus to very minute regions of the brain. We're really happy with RWD's products, and we're really excited to see what they've got in store, their future products that they have in store for the future, and helping researchers worldwide.
Mein Name ist Dr. Heinrich Krollebusch. Ich bin seit 30 Jahren praktizierender Tierarzt und seit 20 Jahren beschäftigen wir uns schwerpunktmäßig mit der Narkosetechnik in der Kleintierpraxis. Uns Tierärzte treibt das Wohlergehen unserer Patienten an, sowie die ständige Verbesserung ihrer Lebenserwartung und ihrer Lebensqualität. RWD hilft uns dabei mit hochmoderner Beatmungstechnik und ebenso modernen Lösungen für Diagnostik und Therapie. RWDs Hilfe können wir Tierärzte sicherer und effizienter operieren, aber gleichzeitig sorgt RWD auch für eine verbesserte Lebenserwartung. Das Wohlergehen unserer Patienten und die ständige Verbesserung ihrer Lebenserwartung und Lebensdauer treibt uns Tierärzte an.
As a national high-tech enterprise, RWD ascribes great importance to technology research and development. Based on 4 technical platforms, including optical imaging, precision transmission, accurate temperature control, and detection of weak signal, we have a grip on leading core technologies across many fields. Since RWD's founding, the company has invested hundreds of millions on research and development, gaining almost 100 patents, both domestic and international, as a result. In addition to a sound manufacturing base, Advanced Processing Assembly Line and Precision Detection System, RWD has built an integrated control system that monitors all aspects of operations from research and development to manufacturing. The name RWD comes from the transliteration of the English word reward, meaning payback to society. Reward is not only the original aspiration of the creation of RWD, but also the continuous belief of the company. In 2016, RWD launched the RWD Preclinical Medicine Enlightenment Scholarship to help with the development of research talent in China's preclinical medicine. So far, the scholarship has been awarded to 38 institutions of higher education nationwide. As the Vice President Unit of the Huxley Wildlife Conservation Society, RWD has donated medical equipment and supplies for wildlife relief and the conservation of the natural ecosystem. In 2020, RWD donated clinical medical supplies worth over RMB 1 million to the Giant Panda National Park to help with safeguarding our national treasure. In the future, RWD will continue to contribute its wits and powers to enhance vital quality and help global customers unlock vital mystery and safeguard vital health.
Hello, dear friends from all over the world. This is Abby from Reward Life Science. Welcome to our webinar. Today's webinar is held by Reward Life Science, which provides equipment in the fields of life science, animal health, and clinic medicine with 23 years of development history. So, agenda for today will be going through with the opening and then continue with the presentation by Professor Manuel for 40 minutes. After this section, we will have a Q&A for 20 minutes. During the presentation, you are welcome to submit your questions in the question box, and Professor Manuel will choose some questions to answer in this part. Due to the time limits, this part will be in 20 minutes, so don't hesitate to participate in this part and ask questions. We look forward to your joining in the discussion. Now it is a great honor for us to have Professor Manuel for today's webinar. Professor Manuel is from University of California, Davis. Research in his lab aims to identify the cellular and molecular mechanisms that regulate arterial smooth muscle excitability and vascular reactivity. Using innovative techniques and approaches, his team focuses on investigating how local and global signals modulate excitation, contraction coupling, and excitation transcription coupling in arterial smooth muscle cells under physiological and pathological conditions such as hypertension, diabetes, and Alzheimer's disease. Let's extend a warm welcome to Professor Manuel. Hello, Professor. Hello.
Hello, thank you for the invitation and the opportunity.
Please open your microphone and share your screen to start. Thank you.
Okay, I hope that you are all seeing the presentation. And first and foremost, I want to thank RWD for the opportunity to present this. Good morning, good afternoon, and good evening to everybody that is in this webinar. Today, I'm going to be talking about unlocking vascular disease connections through ion channel signaling. The goal of the webinar will be to introduce ion channels, the signaling pathway that control the myogenic tone and blood flow, and I'm going to talk about that a little bit. Also understanding how spatiotemporal changes in ion channel regulate vascular smooth muscle contractility and how we can apply innovative instruments such as laser spectral imaging to capture physiological relevance. So the work in my lab focus on the vasculature, particularly in vascular smooth muscle cells. Those are smooth cells wrapping around blood vessels throughout your body. And these cells are very important. The blood vessels are important because they deliver nutrients and oxygen that to meet tissue metabolic demands. They're formed by veins, capillaries, arteries, and arterioles. And those blood vessels have endothelial cells in both arteries and capillary networks, and vascular smooth muscle cells mainly in the arteries and arterioles. And those smooth muscle cells, if we do a cross-section of these blood vessels, will look something like this. This is the medial layer of the blood vessel, and that's where the smooth muscle cells are located. Endothelial cells are lining the interior of that blood vessels. And we can isolate those cells. They look like these spindle-shaped cells. Once they are isolated, we can either measure the ion channels, signaling pathway, proteins that regulate those ion channels and the cell function. And we can also look at mRNA protein levels and so on. And the goal is to go from the single molecule events all the way to the physiology or the pathophysiology of the animals. In my lab, we focus on trying to understand how different diseases affect these vascular smooth muscle cells. And this is a chart of the number of deaths by risk factor in the world, 2017. So this must be updated recently. And in particularly, we focus on three diseases: high blood pressure, hypertension, or high blood glucose level, or hyperglycemia, and obesity, which are actually major underlying factors in diabetes. So those are the diseases. They have high prevalence or risk. They're high risk factors for different diseases and for death, and that's why we're studying it. So let me tell you a little bit about this. Diabetes is a devastating disease affecting millions worldwide. Has a major economic impact not only in the United States but around the world, and it's a disease that has a major impact on the vasculature. and it's underlying vascular complications that causes major causes of morbidity and mortality, such as retinopathy, renal failure, hypertension, and reduced cerebral blood flow that leads to impaired cognition. And hypertension is a high blood pressure. So it's basically estimated half of the population in the US and may suffer from hypertension. It's a highly prevalent disease around the world, especially in Asia. The economic impact is, at least in the United States, around $50 billion, and it could be double or even triple around the world. So it's a primary risk factor for cardiovascular diseases such as heart attack, renal dysfunction, stroke. And one of the things that basically link these two diseases is that they affect the vasculature and in a way impact what is known as this autoregulatory process that helps protect your organs from damage. And that autoregulatory process is known as the myogenic tone. So let me show you a little bit about the myogenic tone. This is an intrinsic property of vascular smooth muscle cells to contract in response to increases in pressure. And that's going to be shown here. This is an artery that's cannulated, cartoon here. This is the cross-sectional area. And what I'm going to show you is the pressure, the diameter of the artery, and then how membrane potential of the smooth muscle cells and the calcium within those smooth muscle cells changes. So what you're going to notice is that with an increasing pressure inside the intraluminal pressure of that artery, the diameter of the artery under normal conditions will distend because of the elastic properties of the artery, but then it will come back down in order to maintain constant diameter. And that's basically what is known as the myogenic response, pressure-induced constriction. And over the year, that myogenic response has been associated with membrane depolarization of the smooth muscle cells that causes an increase in calcium. And that happens because the intravascular pressure depolarize the smooth muscle cells through activation of different ion channels. That increases the open probability of this L-type calcium channel, CAB1.2, that will increase global intracellular calcium and will cause a contraction that you can see here in the diameter of the artery. However, and for the last many years, how hyperglycemia, diabetes, and hypertension alter the L-type calcium channel has been the subject of a lot of study. And by the way, and some of you may realize that these L-type calcium channels are main target of antihypertensive drugs, calcium channel blockers. So that's another reason why these L-type calcium channels are important. And we'll talk a little bit more about these. So these are the L-type calcium channel. The primary the structure of the channel has basically many subunits. The alpha subunit has the pore-forming subunit, beta subunit, alpha-2 delta subunit, and a gamma subunit. They are expressed in many excitable and not excitable cells. They help with muscle excitation, contraction, coupling. In fact, the heart, that's how it depends highly on this ion channel. They contribute to cell excitability, gene expression, proliferation, and migration. And as I mentioned, they're major targets of antihypertensive drugs, and calcium channel blockers are highly prescribed drugs. So we know that this L-type calcium channel can be regulated by many signaling pathways. So G protein coupled receptors that are targeted to GS could regulate the calcium channel, as well as G protein coupled receptors that are targeted to GQ. And one of the key questions that we want to understand is how those processes occur. And it's not difficult. Well, it's apparent to think that likely phosphorylation of the channel in some way might be underlying the regulation of the calcium channel, not only in some muscle cell, but in other cell types by the activation of these signaling pathways. And in fact, that has been shown before. So this is the structure of the calcium channel, and several phosphorylation events that are coupled to GS or GQ signaling has been described. In particular, I wanted you to focus here on this serine 1928 site that is at the carboxy terminal of the calcium of the channel. It was described many years ago by Bill Catterall. That could be it's a readily phosphorylable site. This is actually a cardiac lysis treated with pKa, and you can see the increase in the phosphorylation of that site. if they treat it with cyclic AMP, they also see that increase in phosphorylation. There's also evidence that this site, serine 1928, is a substrate for PKC phosphorylation. And in this study here by the group of Stephen Marks, they showed that actually they stimulate PKC with PMA. They can increase the phosphorylation of that site. And then if they block PKC, then they can bring that down. So this site seems to be a hub for regulation of at least the vascular calcium channel. And that's one of the key questions that we had when we started. So whether this site could be a target for pKa and PKC phosphorylation. And here what I'm showing you is actually human arterial lysates that we stimulated with high glucose. And we saw that the phosphorylation state of that serine 1928 was elevated in response to the hyperglycemic conditions. And we know that that is going through pKa, because if we put a pKa inhibitor, RP cyclic AMP in this case, high glucose failed to potentiate that phosphorylation state of serine 1928. Likewise, we have some evidence that shows that if we take wild-type arterial lysates and we treat it with, in this particular case, angiotensin is a vasoconstrictor, potent vasoconstrictor that acts through PKC. There's an increase in serine 1928 phosphorylation. You can see these two examples that I'm showing you here. So with that, we kind of, and some other data that we have, we kind of conclude that this increase in serine 1928 phosphorylation by pKa and PKC signaling, suggesting that indeed that site, at least in the vasculature, is a substrate for these signaling pathways to be activated. So then we have the question of whether the phosphorylation is structurally and functionally relevant. And first, we started by looking at basically the distribution of the calcium channel at the very super resolution level. So for that, we use this storm microscopy that allows our lateral resolution of about 20 nanometers. So we go from like a regular conventional fluorescent signal that has a lateral resolution of about 250 nanometers to very specific signaling, as highlighted by this cartoon here. So we can go from these diffuse fluorescent levels to highly defined structures of in this particular case, microtubules that we're showing here, how they are, how we can then define the microtubules itself. So we use this technique then to explore the distribution of the calcium channel. And for that, we freshly isolated some muscle cells from wild-type mice. And here we treated these cells with 10 millimolar glucose or 20 millimolar glucose to activate pKa, or control an angiotensin to activate PKC signaling. And under these wild-type cells, what we see is an increase in the clustering size of the L-type calcium channels in both high glucose and angiotensin 2 signaling. And then to examine whether this phosphorylation of this site was important, we isolated some muscle cells from this knocking mouse that has serine 1928 mutated to an alanine to prevent the phosphorylation of that site. And what we notice is that either high glucose or angiotensin failed to increase the cluster size of those channels. So with this, we concluded two very important things. Number one, that the spatial rearrangement of CAB 1.2 channels occur to pathological pKa and PKC stimuli or conditions, such as hypertension and diabetes. And I'm not showing the diabetic state just for the sake of time. But more importantly, we found that the phosphorylation of serine 1928 is essential to promoting this superclustering of the channels in response to high glucose or angiotensin 2 signaling. And we know that that same thing happened in diabetes and hypertension. And these are the references that we have looked at. Then we move on to the functional state. Could this phosphorylation event be functionally relevant? And for that, again, we isolated cells, freshly isolated cells, but then we use the patch clamp technique, and we measure whole cell currents. So the black here and blue are the control currents that we get if we stimulate the cells with this voltage pulse that you see here. And when we stimulated the same cell with high glucose or with angiotensin, we see this potentiation of the calcium current in these cells. However, if we use cells from these knocking mice, the high glucose and angiotensin now fail to basically potentiate that calcium current. And those were exciting results for us because it highlighted that phosphorylation of this serine 1928 site was necessary for both pKa and pKc regulation of the calcium channel. So some lessons that we have learned from that and going a little bit deeper into the functional part of this lecture here. We wanted actually to provide more evidence about the physiological relevance of the observations that we were seeing. So in other words, how we could go from elevating glucose to L-calcium current. And over the years, we have actually described this mechanism in which elevated glucose actually, in this particular case, seems to actually be transported into the cell, metabolized. ATP is produced, and that ATP basically get rid or gets moved out into the extracellular space of the cell, where they activate this signaling pathway that ultimately increase the phosphorylation of the calcium channel, causes this increased clustering of the channel, and increases global intracellular calcium. And that increasing global intracellular calcium not only increases contraction, but also causes changes in gene expression. And one of the things that actually, after we saw this pathway, that we were looking at is what is happening here? How could this be regulated? And this is a study that just got published in Circulation Research. This is the QR code. I'm going to leave it there for a few seconds so that if you want to take a picture. But then we wanted to understand what is happening here, how these ATP molecules could be transported out. And we thought about pannexin. Pannexin is a large pore channel. It's considered a large pore channel or transporter that could actually help facilitate the transport of ATP from the intracellular side of the cell to the extracellular milieu, where it can activate this signal. So we wanted to know about that. I'm not going to tell you a lot about this path, how this pannexin protein is involved in this pathway, but I want to show you something because the research in general was made possible by the development of this GRAB ATP extracellular sensor. And this sensor was developed by Dr. Yulong Li from Peking University. And I know that Dr. Li was also a speaker in this webinar series. So what Dr. Li did and his team did, they developed this GRAB ATP, extracellular ATP sensor, using as a backbone the human P2Y1 receptor. They put a circular permutated EGFP that under basal conditions with low ATP levels, the EGFP is not fluorescing. However, when ATP increases, that causes a conformational change of the P2Y1 receptor, and that causes the CE EGFP to basically fluoresce. So we put that sensor into culture smooth muscle cells. And this is how it looks like. So this is 5 millimolar glucose basal conditions. When we added high glucose, you see that bump in the fluorescence. And 100 micromolar ATP is the control conditions. So it can be better seen here. And what we wanted to know is whether high glucose actually increase extracellular ATP accumulation in these cells. So we are able to track the fluorescent signal from those cells. And what we found using inducible muscle specific pannexin-1 knockout animals is that under control conditions, under the flux animals, the high glucose seems to show a significant increase in extracellular ATP accumulation, as you can see in this red trace here. The 100 micromolar ATP is our control conditions here. However, when we use the inducible pannexin-1 smooth muscle specific knockout cells, high glucose now basically reduce or the knocking out of the pannexin-1 channel basically significantly reduce that glucose-induced extracellular ATP accumulation. So these results for us were exciting because, number one, they suggested that high glucose induces localized ATP release. But number two, it highlighted that pannexin-1 was very much involved in that process. So again, I'm not going to go over all the data that we have related to that project. This is the QR code that you can take a snapshot. But one thing that I really want to show you is whether this signaling pathway is physiologically relevant. And for that, we first start studying basically pressure myography in resistant arteries. These are in vitro studies, okay? And what we have here is we are able to isolate arteries. In this particular case, we're using cerebral arteries as a model system. And we can put them in these glass pipettes, cannulate them, and then with this recording chamber, we can increase or decrease the intraluminal pressure of that artery. So all of these arteries were pressurized to a different intravascular pressure. This is actually a movie of how that looks like. It's not such an exciting movie because it takes time for the artery to develop tone and then responds to these changes in intravascular pressure. But you can see give you an idea of how this works. So when we did that experiment and we did it at different intravascular pressure, what we noticed is that the diabetic flux, pannexin-1 flux arteries, these are the control arteries, develop more myogenic tone, more pressure-induced constriction than the control arteries. And that you can see it here, how this artery constrict more than that is from the diabetic mice than this artery that comes from a sham mice. And this is the amalgamated data showing how myogenic tone is more elevated in those cerebral arteries from diabetic animals than on the controls. However, when we repeated the same experiment, but now using an inducible submuscle-specific pannexin-1 knockout animals, what we found is that these diabetic arteries now didn't have increased myogenic tone. In fact, the myogenic tone was exactly similar to that shown under sham conditions or to those seen in the pannexin-1 flux sham mice as shown here and here. So for us, again, very exciting because removing pannexin-1 seems to prevent the remodeling in the myogenic response during diabetes in these arteries. But then again, these experiments were done in vitro. And one of the goals that we had were basically, can we see this in an in vivo setting? And just to provide more physiological relevance. And for that, we basically turn our attention to laser speckle imaging, and that allows us to do in vivo studies. So laser speckle or laser speckle contrast image is a non-invasive optical technique that measures blood flow by analyzing the random interference pattern or speckle created when the coherent laser light scatters from moving red blood cells in the tissue. And when blood flow is high, the speckle pattern changes rapidly due to the increased particle motion, resulting in lower contrast, while slower flow produces higher contrast speckle pattern. And this relationship between speckle contrast and blood flow velocity allows real-time mapping of microvascular perfusion across different tissues. So this is what we see in the system, and most of the time, of course, we represent the data using the speckle flow index map. Full disclosure, I do have a laser speckle that was acquired from RWD system. I have the RF LS L5-1 in my laboratory. This is actually the setup that we have in the laboratory. So you can see it's occupied very minimal space, and it's easy to have in the lab, and RWD certainly has other system as well. So we have used these systems to study how changes in blood flow occur during diabetic hyperglycemia. And remember the pathway that I showed you before. one of the goals that I have for this presentation is to show you that indeed, if we disrupt different part of this pathway, we can actually see changes or disrupt changes that will normally occur under normal conditions. For these experiments, we actually did some cranial windows. We put a head plate to maintain the cranial window stable, and we removed the skull as well as the dura of these in these animals, so that we can actually add the different high glucose conditions or any drug that we wanted directly to the brain. This is how our setup looked like. And this is actually an example of one of those cranial windows. And in a minute, here we're showing a vasodilatory mixture. And you will see how the artery, how the color changes, right? Suggesting more blood flow going through those arteries. And as well as the diameter changes, so as one would expect. And we have developed some tools that allow us to basically look at the diameter of those arteries and correlate it, not only the changes in blood flow with the changes in diameter as expected, as one would expect. So this is the data that we have collected so far, well, that we have published before. In this particular case, this is a control animal where we have, wild-type animal, where we have 10 millimolar glucose conditions. This is what we were looking when we added 20 millimolar glucose in that artery, and consistent with the in vitro studies that I showed you before, we noticed actually a reduction in the diameter of those arteries and a decrease in blood flow. And we could quantify those things here. And the percent of myogenic tone was elevated, which is in response to 20 millimolar glucose, which is consistent with in vitro studies. And with that increase in myogenic tone, we notice a reduction in flux, again, expected there. If we actually, in this particular study that we published a couple of years back, we removed the scaffold protein ACAP. And what we notice, again, is that under control conditions, as I showed you before, 20 millimolar glucose reduce flow in these animals. But that reduction in flow is completely prevented in the ACAP5 knockout animals. And here I'm showing you a zero calcium and nifedipine just to show you, and we use these basically to completely dilate the arteries and be able to normalize the artery. So that's like that's one approach or technique that we use to try to normalize these examples in a little bit better way. And more recently, from that circulation research paper that just published, we removed pannexin. And here I'm showing you some examples of diabetic animals in this particular case. So the flux animals shown on the top. What we did is we used a zero calcium with vasodilatory mixture to completely dilate the arteries, and then try to measure myogenic tone. And what we found is that diabetic arteries or from diabetic mice, or the artery from diabetic mice, have higher myogenic tone, are more constricted, and therefore has less flux in the brain than arteries from the sham mice. However, if we when we did the same experiment, but in this time using the inducible smooth muscle specific pannexin-1 knockout animals, we didn't see those differences. So diabetes did not affect the myogenic response in those animals, and flow was exactly the same. So this is a way for us to use laser spectral imaging to link mechanisms to phenotype. And this will help deliver in vivo corroboration that these pathways that are perturbed during different pathological conditions are actually relevant under normal conditions. So let me just take you back one more time to hypertension. In hypertension, I show you some data at the cellular level. But here I'm going to show you some in vivo relevance. So and particularly how angiotensin 2 affects blood flow. So the way that we think about angiotensin 2 affecting blood flow is basically it activates this AT1 receptor that activates PKC, and that PKC stimulates the trafficking of some channels to the membrane, but also phosphorylate the channel at this serine 1928 position, where it actually stimulates the superclustering of the channels and the more calcium coming into the channel to cause constriction of the arteries. And in this particular case, one way that we tested the in vivo physiological relevance of that pathway was using mesenteric arteries from mice. This particular example here, these are isolated mesenteric arteries. But what we can do is we can get the mouse, anesthetize the mouse, and remove the endothelium out. We can spread the endothelium so that we can separate the artery, which is like the very thin strand that you see here, versus the vein. So that way you have a full separation of flow between the artery and the vein. And not sure if you can see, this is a movie that's supposed to be running at the moment. I'm hoping it is, yes, it is running, but it's not showing that much of a major difference there. But if we track that movie over time, this is what you're gonna look like, what you're gonna see. Here, what we did is we're plotting the normalized blood flow over time and the normalized diameter over time. And we added different concentrations of angiotensin II. And what you can see is here we're using both wild-type animals and serine 1928 tunnelin in mice. So both are located here. And what you're seeing is that while the wild-type animal that is in blue basically constrict right after we add, the angiotensin, it actually reached a different plateau level than the knocking animal. So both are constricting at the beginning of the addition of the angiotensin. However, one is holding the constriction, whereas the other one is losing that constriction. And that is that has direct correlation to the changes in blood flow that we're seeing, and you can detect it here. And with this type of data, what we can do is we can generate these 3D plots. This is the diameter, the blood flow, and the concentration. And we did these plots both at the peak of the angiotensin response, so here or here, as well as 5 minutes after we treated that, in other words, here. And what you can see from this data is that if you look at the peak only, there's not that much difference between the Ang2 effects in the wild type and the knocking animals. However, when we look at the steady state level, you can see the difference here. It shows that the knocking animals actually have more like a reduced diameter, reduced flow compared to the wild type animals. And that is important because that's what we would expect by adding this Ang2 signaling. And it reveals that the phosphorylation of this serine 1928 site is extremely important for maintaining a higher level of constriction and lower blood flow in when those arteries are stimulated with angiotensin II. And it shows this key role for serine 1928 phosphorylation in modulating arterial diameter and blood flow in vivo. Now we have used this technique for other practical purpose. This is actually a mesenteric sample here, and we are adding high angiotensin II, the vasoconstrictor that I showed you before. So this image perhaps is better than what I showed you before. But this is how blood flow and diameter changes in mesenteric arteries in response to Ang2 signaling. We have also used like the laser spectral image to study sexual dimorphic responses in adrenergic stimulation. And here I'm showing you male and female arteries and how isoproterenol, a beta-adrenergic signal, basically causes dilation of the arteries more in female than in males. And that increased dilation is correlated with more blood flow in females than in males in response to this isoproterenol signal. So we can apply this technique to different animal models. We can look at progression of vascular disease and also apply it to different preclinical studies. Let me just give you one more example of how we have used this technique. This is the mouse paw approach using our system in the lab. we started to implement this PAW configuration because of its accessibility, its reproducibility, and sensitivity to different techniques. So what I'm showing you here is like the PAW pathway before we added or we injected the mice with epinephrine. And once we added the epinephrine in the mice, you can see this reduction in flow. Epinephrine is a vasoconstrictor. So it constricts, you're going to have a reduction in flow that can be detected there immediately. And that's what we're seeing in these trace that I'm showing you here. So these experiments are useful for assessing diabetes and metabolic syndrome impact in the microvasculature, ischemic reperfusion studies, drug testing and intervention. And we have done one of those drug testing and intervention recently with a collaborator here at Davis. That is super exciting. And we can also study inflammation and immune activation. So I hope that you have shown you how to leverage laser spectral imaging to gain insight into physiological relevance of mechanism regulating ion channel function and how those changes translate to physiopathological responses. And what I show you, I hope that I have provided enough information to convince you that during these pathological conditions of diabetic hyperglycemia or angiotensin, activation of these GPCRs basically seems to converge on serine 1928 phosphorylation on the calcium channel to hyperactivate the channel and increase the clustering of the channels. With that increasing clustering of the channels, there's an increase in the calcium amplification that occurred, the contraction of the smooth muscle cells. There's changes in gene expression that I didn't show you, but are happening. And that will have a tremendous impact on how blood flow and blood pressure are regulated. We see this serine 1928 phosphorylation event as a potential target for potentially developing new therapeutics for hypertension, diabetic vascular complications, as this is basically the increased phosphorylation of this site seems to be a risk factor for cardiovascular complications. So to conclude, I think ion channels rock. They play key roles in regulating resistant arteries. Much more there is to discover in terms of ion channels and signaling proteins. And perhaps importantly for us is that you get the idea that laser speckle is a powerful tool for assessing microvascular changes into real-time pathophysiological evidence, and it's a potentially is available technique, should be an available technique in every lab. So with that, I'll stop here. I would thank the team that has done the work, particularly Miguel Martin Aragon, Jay Taylor, Eric Silva, Nuria Sayaman, and Yung Hong when he was in the lab. The different collaborators, and I would not like to leave without saying thank you to the National Heart, Lung, and Blood Institute for supporting our research and the American Heart Association. This is the team over the years, and with that, I'll stop here. Thank you all for listening, and I'll happy to take any questions if there are any. So I do see a question in the Q&A section, and it is, I'll basically say the question. So is high glucose induced ATP release dependent on Pannexin-1 channels? Does it release mechanisms involve vesicular exocytosis or volume regulated ion channels? Yeah, this is an excellent question. And the answer is Pannexin. We think that Pannexin-1 channels are involved. It's not the Pannexin-1 channels are considered to be ion channels. The way that I'm starting to think about Pannexin channels is more in the context of a transporter. because it doesn't make too much sense to have an ion channel that could open and close at all time, such as the pannexin channel. The membrane potential of a smooth muscle will not be controlled. So in that regard, I think it's more of a transporter. There's some evidence in the field that that could be the case. Whether there is an involvement of vesicular exocytosis, I think, again, it's not that much exocytosis, but more of pannexin 1 transporting ATP out of the cells. However, I will say that we haven't discarded whether volume-regulated ion channels are involved. They might well be involved, and we have some evidence that that could be the case, particularly when there is increased osmotic pressure. Those volume-regulated ion channels may contribute. The important part, and I think that the critical part here will be that we don't have evidence yet that the volume-regulated ion channels are within that complex that I show you in multiple slides. We do have evidence that the pannexin 1 channel is within the complex that activates the purinergic signaling and the pathway that I show you. And in that sense, I think that this is a very local signaling pathway that gets activated. And whether volume-regulated channels are involved remain a subject of investigation. We have another question here. Does CAB 1.2 channel clustering have long-term structural implications in vascular remodeling? We think that they may have. The evidence for that is when some of the studies that we have done have used human diabetic arteries from patients that underwent gastric sleeve surgery. The way that we got those samples here, they travel overnight in a controlled solution, and yet after maybe 12 to 24 hours of traveling, the arteries and the cellular work that we did was very consistent with the results that we were observing in the animal model. So that suggested to us that any basically preventing or moving or getting the samples into a more normoglycemic state was not actually causing an immediate change in the structural remodeling that we were observing, and that this is more of a persistent or a more permanent effect. So there's likely some epigenetic changes that are occurring, and we are exploring that possibility at the moment. The other question is this process associated with cytoskeleton reorganization or membrane microdomain, or such as lipid raft participation? Yeah, great question. It's certainly likely associated with cytoskeletal reorganization. We have some evidence that suggests that the cytoskeleton, F-actin and other proteins are important for contributing to this increased clustering of the calcium channels. Whether lipid rafts are involved, we don't know at the moment. It's, again, an area of investigation in the lab, and hope to have some idea about that later on. Okay, so another question that we have here is, in the comorbidity of hypertension and diabetes, is CAB 1.2 channel function subject to synergistic or antagonistic regulatory pattern? Yeah, great questions. Again, I will say yes. They are subject to a lot of synergistic and antagonistic pathways, and in addition to that, we don't know enough what happened to all of those pathways when the patient basically start giving some treatment option that will prevent or that would act on certain pathways versus the other. So there's a lot of work to be done to try to understand what is happening in hypertension and diabetes related to calcium channel. It is clear that the calcium channel activity goes up, at least in hypertension. That's why it's one of the key targets for antihypertensive drug. In diabetes, the data that we have suggests that it goes up, but maybe later on more advanced effects could be completely different, and we are not unclear how what are the both the synergistic and antagonistic pathways acting on at the moment. So that's where we are. Okay, so yeah, I mean, I hope I have answered your questions, and okay, so I apologize. There's like a few in the chat. I was looking at the Q&A. There's a few in the chat. Does a myogenic response have distinct ion channels based on different vascular beds, cerebral versus mesenteric arteries? Yeah, that's a great question. And indeed, it does have different ion channels in different vascular wells. The distribution of the ion channels, the ion channels could be the same, but the distribution and how they are regulated may be different. And therefore, that is an area that needs to be evaluated. I mean, like there is clear evidence that different vascular beds can be regulated in different ways. And that could be in part because of the different ion channels that are there and how they are regulated. The other question that I have is the molecular basis of sexual dimorphism in vascular response related to estrogen-mediated regulation of CAB 1.2 channels or BK channels? So the BK channels are the large, the calcium-activated potassium channels, large conductance calcium-activated potassium channels. That's a great question, and indeed, it could be related to that. We are currently working on OBX mice in the lab, try to understand how those, whether the sexual or hormones could be influencing how the channels are expressed, how that expression could be altered during pathological conditions, and whether the sex hormones, what is the influence of that. And at times with the OBX treatment options, you will get more questions than answers. There's a lot of variability that occur. So it's not a straight, it's not a black and white type of regulation in that particular case. Other question that I have here is, in pressure myography experiments, how can one distinguish between endothelium-dependent and endothelium-independent vasoconstrictor response? It's a great question. And in this particular case, the way that we ensure that what we were seeing was mediated by smooth muscle cells and not endothelial cells was basically we repeated a lot of our experiments in intact arteries were repeated using endothelium denuded arteries. So the way that you do that is basically you cannulate the artery and you pass a couple of bubbles through the artery, and that basically disconnect, impact, and alter the function of the endothelium. With this technique, you always want to do make sure that you've disrupted the endothelium by adding NS309 or like any drug that worked through the endothelium to cause dilation. So we did that, and we show that when we remove the endothelium or disrupt the endothelium, that NS309 gets basically don't causes dilation of the arteries as one would expect. So that's how we know that the mechanism that I show you is vascular smooth muscle dependent. However, with the laser speckle, then we went back and we integrated and showed that even in that complex environment where you have many cell types working together, including endothelial cells, astrocytes, and other cells, that indeed the high glucose effect that we were seeing in vitro also occurred in vivo. And that was like a good an experiment for us to reassure that what we were seeing has physiological relevance.
Okay, now we are done at the Q&A. Thank you very much for your presentation and answer, Professor Manuel. Thank you for your bringing us your academic insights in ion channels, signaling pathway, and control of myogenic tone and blood flow. We have gained valuable knowledge about ion channels, and it plays critical role in maintaining vascular function. Finally, please allow me a few moments to introduce our company's product, Laser Speaker Imaging System. First, actually, blood changes are important indicators of neurodegenerative diseases and metabolic disorders. So I would like to take a moment to share you that latest method that can further support our efforts in this area, which was highlighted in Professor Manuel's research known as Laser Speaker Imaging System. This solution offers non-contact monitoring of microcirculation without the need for contrast agents, making it safer and more convenient. With this technology, you can easily export pseudo color maps for blood imaging, blood perfusion data, tube angle, and tube diameter. What's more, it provides high-resolution imaging with every detail. All that makes Laser Speaker Imaging System an invaluable tool for advancing our understanding in neurovascular and other molecular microcirculation, whether in AD or other diseases. Here are some cases of laser speaker. Picture in the left is the cerebral flow in the mouse, and the right is the rat. And for the stroke model, clear to see that the blood decreases after the surgery. So it is an even better tool for you to monitor microcirculation. We are very proud to announce that it has been 4 years since the product was launched, with over 200 systems installed in situations and hospitals worldwide, and published in highly rated journals, including Science, Cell, and Nature. We hope our laser speaker imaging system will be greatly helpful for you. Thank you. Okay, all right, thank you very much, Professor Manuel. Due to the time limits, we are done at this webinar, and once again, thank you very much for your time today showing your wonderful work. And thank you for everyone attending and having a great time. Thank you very much for everyone. We're looking forward to following up with you on the webinar next time. Thank you. Bye-bye.
Against all these threats and challenges to life, constant progress of research in life science and tight collaboration in relevant industry chains are essential for achieving better vital quality. Originated in 2002, RWD has been constantly furnishing customers with reliable solutions and services in life science, animal health, and clinical medicine, and dedicated for better vital quality within our wits and powers. Our products have been sold to over 100 countries and regions around the globe and won worldwide customer confidence and support. At RWD, we launched 6 solutions in the fields of life sciences, animal health, and clinical medicine at the forefront of the world, including animal surgery and modeling solutions, which ensure experiment efficiency through diverse animal disease modeling and model evaluation techniques. In vivo imaging solutions, which provide tangible tech support in fields such as biology, pharmacology, materiology, and translational medicine, among others, by accurately showing blood flow and changes in other indicators of live beings in visualized imaging. Neurosignal research solutions, which are valuable tools for neurocircuit analysis, as well as research into mechanisms for neural system diseases, development of brain-machine intelligence technology, and more. Histopathology solutions, which improve the quality and efficiency of pathological diagnosis while sectioning with high accuracy and efficiency. The cellular and molecular biology research solutions, which advance efficient scientific research through covering all conventional experiments, including cell separation and purification, self-culturing, self-analysis, and molecular biology. Animal diagnosis and treatment solutions, which evaluate diagnostic efficiency and treatment efficacy of surgery with anesthesia, dental services, rehabilitation, and physiotherapy, urinary system diseases, etc. And our assistants, science researchers, have published thousands of papers in a number of world-leading academic journals, including Nature and Science. On top of that, many scientific research institutions, medical institutions, and pet diagnosis and treatment centers have attained their success by leveraging our 6 solutions up to now. We will maintain cooperation and innovation with an open mind, boosting growth within many industries through joint efforts with our partners.
So we've been collaborating and cooperating with RWD for quite some time now, and their self-developed products have helped us in the laboratory maintain accuracy and improve accuracy and help our efficiency. We've used everything from cryostats for sectioning various organs of the body, all the way through to stereotactic surgery solutions to deliver small amounts of virus to very minute regions of the brain. We're really happy with RWD's products, and we're really excited to see what they've got in store, their future products that they have in store for the future, and helping researchers worldwide.
Mein Name ist Dr. Heinrich Koddebusch. Ich bin seit 30 Jahren praktizierender Tierarzt und seit 20 Jahren beschäftigen wir uns schwerpunktmäßig mit der Narkosetechnik in der Kleintierpraxis. Uns Tierärzte treibt das Wohlergehen unserer Patienten an, sowie die ständige Verbesserung ihrer Lebenserwartung und ihrer Lebensqualität. RWD hilft uns dabei mit hochmoderner Beatmungstechnik und ebenso modernen Lösungen für Diagnostik und Therapie. RWDs Hilfe können wir Tierärzte sicherer und effizienter operieren, aber gleichzeitig sorgt RWD auch für eine verbesserte Lebenserwartung. Das Wohlergehen unserer Patienten und die ständige Verbesserung ihrer Lebenserwartung und Lebensdauer treibt uns Tierärzte an.
As a national high-tech enterprise, RWD ascribes great importance to technology research and development. Based on 4 technical platforms, including optical imaging, precision transmission, accurate temperature control, and detection of weak signal, we have a grip on leading core technologies across many fields. Since RWD's founding, the company has invested hundreds of millions on research and development, gaining almost 100 patents, both domestic and international, as a result. In addition to a sound manufacturing base, advanced processing assembly line, and precision detection system, RWD has built an integrated control system that monitors all aspects of operations from research and development to manufacturing. The name RWD comes from the transliteration of the English word reward, meaning payback to society. Reward is not only the original aspiration of the creation of RWD, but also the continuous belief of the company. In 2016, RWD launched the RWD Preclinical Medicine Enlightenment Scholarship to help with the development of research talent in China's preclinical medicine. So far, the scholarship has been awarded to 38 institutions of higher education nationwide. As the Vice President Unit of the Huxley Wildlife Conservation Society, RWD has donated medical equipment and supplies for wildlife relief and the conservation of the natural ecosystem. In 2020, RWD donated clinical medical supplies worth over RMB 1 million to the Giant Panda National Park to help with safeguarding our national treasure. In the future, RWD will continue to contribute its wits and powers to enhance vital quality and help global customers unlock vital mystery and safeguard vital health.